Welcome everyone. Thank you to OIS for putting together this spotlight on drug delivery panel. Those presentations were amazing. I thought I'd seen everything, but I've seen some new stuff, so pretty cool. We've got a great panel here. Again, my name is Barry Kuppermann. I'm down the road in UC Irvine, so we're local here. And again, I'll do a brief presentation to give a bit of a drug delivery overview and then have a bunch of questions for this great panel. These are my disclosures, just to list a million, but here are our panelists, Dr. Caroline Baumal, Tom Ciulla, Dr. Shamika Gune, Dr. Peter Kaiser, and Dr. Michael Singer. So welcome all to the panel. And again, I'll just cover a couple things. This is actually a slide that I borrowed from Tom Ciulla a couple years ago, where there's so many different ways to deliver drugs to the back of the eye. We've been hearing a lot about them. Periocular injections, we didn't hear much about that today, but that's been around a long time. Intravitreal injections have been the mainstay, as I'll cover in a moment. Reservoir drug delivery devices, subretinal injections, suprachoroidal injections, as well as topical and systemic agents. We heard a little bit about topical agents today, but a lot of excitement in this whole field. And again, it's worth noting that it was at this meeting, i.e. ASRS, in Montreal exactly 20 years ago, and that's where you see the top at the beginning of that curve, that we all together saw Phil Roosevelt present on Avastin, Joan Miller present the Marina results for Lucentis, and we all bought our first OCT machines, the temporal domain OCT. It was an incredible marriage of technology and pharmacology, and we also, I will say, looked around the room as retina specialists and said, who the heck are all these other people? Suddenly the pharmaceutical industry and the financial industry, the investors were suddenly in our midst, because we were just a random surgical specialty that didn't have a lot of attraction or interest. So the day it started, literally, I don't know what the date was, it was in July of 2005 in Montreal. So that's the beginning of this whole story. And we're still, nobody would have expected that 20 years later we'd still be injecting drugs into the eye. I don't think any of us would have believed that. And part of the problem, and where we have gratefully, and it's been alluded to by other speakers, what happens over time is in clinical trials, patients do well, and then over time, patients do less well. All these are extended duration studies, and we know that we're doing, even with treat and extend, we're not doing as well as we thought we were. We'd want to do this. We want to keep vision stable over time, but unfortunately, under the current methodologies, they deteriorate over time. So there's still a definite unmet need. So the whole point of the drug delivery panel or the presentations we just saw are still very germane and relevant to us. This is a cartoon that was taken from the Port Delivery System portfolio, and that's what that's showing here. But again, when you give intravitreal drugs, you overload the eye, the vitreous, with drug, just to get some drug there 28, 40, 36, 35, 42 days later. And in fact, treat and extend, the mainstay that we use, that there's a drug holiday between each injection. In no patient, really, when you're treating out 12, 16, 18, 24 weeks, the drug is long gone. It's just the disease is not reactivated. And this shows with the drug delivery system, you get more extended at much lower doses. And again, this is where we were back before in the far left, Visiodine, laser photocoagulation. Then I left off Macogen here. That belongs in there before first generation drugs. But then 20 years ago is when we got Avastin. 19 years ago, essentially Lucentis and Eylea. About 14 years ago, second generation Eylea high dose and Vabysmo, a bispecific. We heard a lot about the TKIs today, which are with the Ocular Therapeutics and EyePoint and their drug delivery systems, which again is going to change our paradigm. Also Clearside with the suprachoroidal delivery. And gene therapy we didn't hear much about today yet, but that's where we're going as well. Adverum and 4DMT have intravitreal delivery, and REGENXBIO has both subretinal and suprachoroidal delivery. And again, there's different approaches to ocular gene therapy. Again, as I just alluded to, suprachoroidal, subretinal space, and vitreous chamber, without spending too much time talking about that. But there's different approaches. These are basically, these gene therapy approaches turn the retina into a drug-producing factory. All of them right now are focusing on aflibercept. They're not the gene editing approach where it's a curative disease, where you go to a point mutation and transform the genome. You modify the retina to become a drug-producing agent. So it's a very interesting approach. Additionally, just to conclude with a couple of comments, we heard a little bit about earlier in the interview about cell therapy. So again, there's a recent approval. These for Encelto, which is modified RPE cells. These are ARP19 cells that are in a laboratory. I have them in my laboratory, as we all do. They're hard to kill, and you can modify them. So these are modified to produce CNTF. They can be modified to produce other agents. They're placed in a straitjacket, sutured into the eye, and basically live forever. They keep taking one out every year, and it's still alive, still producing. I've warned them. I said, this stuff lasts too long. It's maybe a lifetime therapy from basically what this is baking the bread fresh daily. It's cells that produce a drug every day. So similar but different than the gene therapy approach. And finally, the last slide for concluding slide before we go to the panel, there's also retinal progenitor cells that are being looked at for retinitis pigmentosa. A company called J-Site, others are looking at this. And these are not really exactly drug delivery factories. These are trophic factories. So these are young, healthy retinal cells that exude all the trophic factors that young, healthy retinal cells do, so they create a healthier milieu, a healthier retinal environment, so they could be very effective for something like neuroprotection, potentially neuroenhancement, treatment of things like GA, dry AMD, or retinitis pigmentosa, which is being tested. So to conclude my part of this, in terms of the presentation, there's still a lot of diseases looking for improved pharmacological solutions. We're really struggling, meaning we're getting great results. We're grateful to all the pharmaceutical companies that have given us these amazing drugs that have changed the lives of our patients, but now we're looking to do better. And you saw the enthusiasm and interest from the presentations earlier today. Again, we're looking for drugs, delivery therapies that are either significantly extended or the potential for one and less. They're trying to get away from the one and done concept of gene therapy, but certainly one and less is where we're trying to be. And again, there's port delivery system, a surgical approach. So there's a lot of enthusiasm, excitement around drug delivery because we've lived for 20 years in this era beyond what we expected with great success, but now we're looking for more. So I'd like to actually ask the panel, and I'll turn to the panel. Peter, this is sort of one I was thinking of you, again, and we saw Sanjay produce Peter's courses at Cleveland Clinic, but all working with Ocular Therapeutics. You have a unique sort of clinical trial design, something we've not seen. Is this a new standard? Is this something that you were encouraged to do? Is this something you'd like to do? I mean, again, you took it a different approach with Ocular Therapeutics than EyePoint did, who did the more traditional, what we're used to seeing, two identical non-inferiority trials. You took a superiority trial and separately a non-inferiority. What are your thoughts about that approach?
Yeah, I mean, there's many ways to skin the cat, and the thing that's important here is this idea of the two trials not necessarily being identical. We certainly could have done two identical studies and really learned nothing how we could use the drug in clinical practice. So that's why SOL-1 is a superiority study, because we really wanted to figure out how long the drug lasts, and that'll help with our label. But in the second study, the goal here was to compare it, obviously, to Eylea 2 milligrams, but we're also comparing it to Eylea HD, and we're going to be able to see over time rescue rates, fibrosis rates, visual acuity outcomes. The idea that you need to have the two studies be identical is false. The FDA just cares about two well-controlled and masked clinical studies. So doing it either way doesn't matter. Think back to Marina and Anchor. Those are two totally different studies if you think about it. One had no control and the other one had PDT as a control.
Fair enough. So what about, so that you lose the ability to pool data, et cetera, but you also have these two studies that sort of answer different questions. The FDA was obviously receptive to this.
The FDA doesn't. The FDA is always allowed two different studies. They've never required two identical studies. If you think about it, the reason why when the VIEW study was designed, and I was part of that, we had to compare ourselves to something. We couldn't do what Lucentis did. We couldn't go against sham like we did in Marina. We had to go against Lucentis, and that's the whole idea of a non-inferiority study. Every study thereafter did that, but the FDA began to become upset, in particular with this idea of the disease activity assessment. So as we started to try to differentiate sort of from Eylea 2 milligrams by having this disease activity that was only applied to the new drug and not applied to Eylea, the FDA was saying, well, you didn't prove that you actually last longer than Eylea. And that's why the guidance came out saying, basically, at least in CEDAR, we don't want that type, or CBER, sorry, we don't want that type of drug trial in the future.
So again, that raises some interesting point, but it occurs to me what's been alluded to is potentially with extended drug delivery, a lack of fibrosis, and there was this potential concern about chronic delivery might lead to expanded geographic atrophy. I know, Shamika, you've been working with the port delivery system. There's some data from there that shows there's actually potentially less geographic atrophy. That's not a question I thought of earlier, but based on some comments earlier, thoughts about that finding, which was a relief. I mean, there had been this debate early on in anti-VEGF era because the second most important effect of VEGF as a molecule is it's a survival factor. So if we're giving anti-VEGF injections, could we be promoting geographic atrophy? And again, we found the opposite with chronic drug delivery.
Yes, it was a really, we were very encouraged with that finding. We went in to really see whether, because there was a concern that continuous delivery, we might see actually the opposite. We might see more atrophy given the evidence on macular atrophy thus far. But actually what we found was that, the progression of atrophy in the control phase when we are comparing with monthly injections is looking much better when you look at the PDS arm. And we have some hypotheses, whether it's related to the pulsatile, the spike in the IOP, or just being able to control, the expansion contraction of the retina, whether that's protective. But I think maybe a little bit more work needs to be done as to why, but I think it's definitely encouraging to see that when you deliver drug in a sustained manner and avoid those peaks and troughs, you are potentially also minimizing the progression of.
That's a great relief to everybody in this field. Michael, you've got some
So
comments.
it's interesting. So it's funny. I'm thrilled that they've proved this thing, because 4 or 5 years ago, I went to Genentech and said, I'm a simple guy. So if you get pulsatile injections, it's kind of like hitting your hand. Eventually, your hand gets red and irritated. If you stick your finger on your hand adapts. And if it gets red and irritated, it obviously causes inflammation. And what do our GA drugs do? They actually, with complement, they target inflammation. So it's wonderful that it actually proved the thought that given sustained delivery is a lot more better for the eyes and better for the homeostatic environment.
Thank you. Let's move on to the next question. Gene therapy. It's an emerging field. There's various delivery routes. Tom, you've been involved in this in various ways, certainly in the past. Your thoughts about the various pros and cons of subretinal versus intravitreal and suprachoroidal approaches, which are all in development right now?
Yeah, it's a great question. So yeah, I was at Spark and supported the launch and approval of Luxturna. Then I was chief medical officer at Clearside and worked on suprachoroidal therapy and supported the approval of Xipere. And so they all have their pros and cons. I mean, subretinal is obviously has regulatory precedent with the approval of Luxturna. We're basically putting the gene therapy on the layer that needs to be transduced. We're going to have high drug levels produced locally. It's an immune privileged space. There's less risk of inflammation. Of course, it is somewhat invasive. I think Furious pointed out earlier that subretinal is being adopted by our community. Our community always adopts innovation, and I'm sure that will continue. Intravitreal obviously is the easiest solution in terms of access. It democratizes access to gene therapy in some ways. However, it does have the highest risk of immune response. You have the internal limiting membrane, which reduces access to the retina when you deliver intravitreally. There are various companies working on optimized capsids to penetrate the internal limiting membrane. So I think eventually that problem will be solved and it will be a viable approach. Suprachoroidal is another office-based delivery method, probably has less inflammation than intravitreal. It does require a device, so for those of you who develop gene therapies, you potentially have an optimized capsid, you have a new device, and then you have your therapy. So it adds a lot of complexity to the development. But I think suprachoroidal will be viable as well. Regenexx Bio is doing those studies now. Their diabetic retinopathy study with suprachoroidal delivery of their asset looks, quite promising. So I think each of these modalities will have a place in the future. And again, they all have their pluses and minuses.
Caroline, your thoughts on this same topic? It's again, I'm going to ask you a couple questions. I'll get to GA with you because of your other hat, but as a retina specialist, what excites you about gene therapy? What concerns you? What are the pros and cons from your perspective?
Well, I think the biggest thing to complement what Tom was saying was that it really depends on the disease that we're treating. And for example, subretinal gene therapy, which retina physicians will get, that will get better and people will adopt that, it might not be the appropriate therapy for geographic atrophy, for example, but it is more appropriate, certainly for younger patients. So it really depends on the disease that you're treating and who's going to be a good candidate. So I would really take that into mind. I mean, I think a lot of physicians would like a gene therapy, a more one and done, but there are also risks with gene therapy, with giving it to young people. Are there any long-term risks? The development takes longer. The standards for safety are very high, so we have to take that all into consideration.
Now, you've raised an excellent point. And again, this concept of one and done versus what I like to prefer is one and less would be good enough for me. Peter, your thoughts on gene therapy and the decreased, again, do we really need it to be a single shot treatment, or is it, if we do initial therapy and then a supplemental injection every year or two, is that a failure?
Well, I mean, it depends on what it costs. So,
Yeah.
so if it costs $500,000, then you have to repeat it every couple of years. I think insurance companies aren't going to cover.
No, it would be a supplemental injection of a free drug, I think, with the gene therapy
Well,
in
again,
the
it
background.
depends on the cost,
Yeah.
right? Everything depends on cost. I mean, and the interesting thing, what Tom brought up is intravitreal is by far the easiest, but it's also the most inflammatory. And so to avoid that, you reduce the dose that you apply, but that also reduces transfection rates.
Right.
So the yin-yang, right? At the beginning, if you look at this, you say, well, subretinal is the hardest, intravitreal is the easiest. But like anything, we never thought we'd do intravitreal injections like we're doing now. Maybe we'll be the same way with subretinal in the future. We just simply don't know. It's whatever is the safest of these three methods with the best transfection will win.
So, Mike, what do you think about the cell-based therapies? There's Inceltone now available. You're looking at it for your MacTail patients. And again, it spits out CNTF, which is a wonderful neuroprotective agent. I think I've encouraged them to start making some money off of this and then start looking at other indications like geographic atrophy or dry AMD. CNTF is a wonderful neuroprotectant. It could be a wonderful therapy. Your thoughts about cell-based therapies?
I think cell-based therapies are fascinating. I think they really, they've been doing this a long time. You heard this morning the history of the company. I mean, we were involved when they used to have an aflibercept-based cell-based therapy, and I think that would have worked had they had a higher dose that went off. I think this is a very interesting technology. I think Peter's point really does hit home, though. I mean, obviously for an orphan disease, they can have a very high price tag. As you do a more common disease, then it becomes a limitation. But I do believe that this is a way that you potentially can get one and less. The one and done is a greater problem, and I think the question is, the less becomes the issue. Because what we don't know with all these great therapies is we don't know who are the people who are going to need supplementation and how do we screen for those people. In DME, it may not be a big deal because you can let it sit a month or two, but in wet AMD, it becomes a bigger process. Now, obviously, home OCT will make a big difference, but how does that integrate into an individual practice's data stream, how they do their day-to-day thing? Do you jump up and down when you see a difference and have the patient come in? Or do you wait a little bit for a couple of days? So I think the logistics are where the devil's in the details.
Hey, Barry, I think when we think about cell therapy, it's a little bit of an oversimplification. I think of cell therapy as sort of in two different flavors. There's neurotrophic cell therapy, and there's replacement cell therapy. And when we started off this panel talking about sustained release, so I think on the innovation spectrum, I think sustained release is in the near term, gene therapy is in the midterm, and cell therapy is in the longer term. And I think cell replacement therapy is even longer term. It's probably not ready for prime time. I think the benefit of cell therapy is the neurotrophic aspect of it. But I also think that can be achieved with gene therapy. And then the question becomes, which is simpler and more reproducible, gene therapy with neurotrophic factors or cell therapy with neurotrophic factors? I suspect cell therapy with neurotrophic factors is probably a little bit further out on the innovation spectrum because the development is so complex and probably the costs are so high.
No, I agree completely. Again, when I refer to cell therapies here, this is still a drug delivery panel. So those replacement, that's a whole different area that has nothing to do with drug delivery at that point. We're rebuilding the retina. That's still pie in the sky of the future. So we're looking to that. So again, Caroline, we're now in this era, still this comfortable era that we've evolved over the last 20 years, that we started off with all the data from clinical trials, and we started dutifully injecting every 4 weeks, and then we started playing with PRN, and then we evolved to treat and extend, which most of us have embraced. And so the nice thing about treat and extend is for the last zillion, 20 years, but let's say it took 5 years to get to treat and extend and evolve it. Last 15 years or so, we have been perfectly matched between our exam dates and our treatment dates. It's a marriage made, and we also get the OCT, we confirm the lack of fluid, we examine the patient, and we give them their injection, say, see you back in X number of weeks, whatever their number is. Now with extended drug delivery strategies, what is going to be, so now let's say we've got a drug delivery system like Ocular Therapeutics or iPoint, that will last, we'll have to figure out, is it 6 months, is it 9 months, is it 12 months, how variable it be? Then we've got the gene therapy approaches, what does that mean? What is your view of the future and how we're going to manage our patients? How are we going to match the exam interval with the treatment interval?
Well, for what I think we're going to get, and I hope the TKIs come to fruition for all of us. So we have another MOA. I mean, we've seen that with Vabysmo. Another MOA added into the mix is helpful. So what I think that we're going to learn a lot from this superiority study. We'll see how long that lasts. But I think that we're going to basically have a safety for our patients. I mean, treat and extend, it's not that there's science behind the treat and extend, but it allows us to identify patients that need to have a lot of treatment. Those are the patients we see every 4 weeks and they still have fluid. Okay, this is patients going to need a lot of anti-VEGF versus the patients that we see, they only need a few anti-VEGFs every once in a while. So somehow it's letting us to distinguish what the patient factors are because we don't know how the genetics and the specific patient factors are. So that's what treat and extend allows us to do. And now we're going to have a safety net, especially for the patients who need to have a lot of injections. Like, hey, we'll give you this, and if you don't show up exactly at 4 weeks, that's fine. So I think that that's going to help us. But ultimately, all of the things that we're doing now are talking about modifying the disease. And I think this was what Tom was getting a little bit to. We're giving them modifiers. We're not curing the disease. Like, once we can find a cure, then we'll all be on this panel talking about something completely different.
So, Peter, what are your thoughts? In a TKI drug delivery system world, do we just bring them back every 6 months and inject them again, or is there more nuances to it? What are your thoughts?
Yeah, I think you're right. I mean, remember, treat and extend was designed because none of us wanted to do monthly or every other month dosing. So we said, well, we're not doing that. Let's do something different. And that's where treat and extend came. The TKIs may offer something very interesting, which is you look at, for instance, Ocular Therapeutics. We'll know if it lasts 9 or 12 months. But, I'll call it the Ozurdex problem, right? I want to know when I need to see that patient back. Or the majority of patients, I say. Maybe not 100%, but if it's a majority of patients can make it a certain amount of time, then I could tell the patients, look, I want to give this True 6. We're going to do this every 6 months, and you're going to be fine. There'll be a few patients that need extra injections, but that is something that patients really want. As physicians, I can reliably do it. If you look at the port delivery system, they were smart to do refills at 6 months because that's when I want to see my patient with wet AMD. I'm not going to let them go a year. That's way too long to not know what's happening, especially in some of these older patients. So, 6 months may be the sweet spot, at least for now, in terms of how we treat the patients.
Agreed. Shamika, so your thought, I was going to ask you to, like on this following, the last question, with your port delivery system, how do we design, rethink endpoints, patient selection, et cetera? But please feel free to comment on what you were going to,
Yeah.
but the next question is yours as well.
Thank you. Yeah, just a quick, I think, as these, newer therapies that are under investigation, they read out their data, especially over the longer term, that will help define, how to monitor the patients. For example, as Peter said, with the port delivery system, we know 95% patients are not going to need a treatment, right? So then that helps you sort of anticipate, what is going to be their lowest interval, and then if, just generally helps you plan for when to bring them back. And I think the longer data from all of these additional agents is going to help define the intervals for that as well. In terms of your question about what additional endpoints, of course, BCVA-based endpoints are not going to go away. They are the gold standard and are needed. And of course, durability endpoints, specifically the ones where, you're demonstrating durability without compromising on the efficacy. But I think given sort of the environment that, once it's one thing to get it approved, but then the next challenge is to get it implemented in clinical practice. And I think for that, you do need, endpoints beyond the clinical endpoints. You need patient-reported outcomes, which is tremendous in terms of just creating belief, both from the treaters as well as those that are, who you're offering the treatments to. And then many times overlooked are health economics-related endpoints, because ultimately payers need to pay for these treatments. So I think it would be smart to incorporate these additional endpoints, as you're developing the product to help quicker adoption after it's approved.
Thank you. So Michael, in the era of GA, here we are. What are your greatest unmet need for, from a drug delivery standpoint in our patients with geographic atrophy with current therapies?
I mean, it's pretty obvious that visual function is what it's all about. And although we do believe that slowing the disease is very important, and it definitely in the long run probably translates to something, we can't measure that something. And I think that's why there is such a number of people who do not do complement inhibitors, because they can't get their hands around visual function analysis. And we need lots of ways of looking at different visual function. I mean, Shamika says, right, best corrected visual acuity will be there forever. But again, these people, there are many of you who are physicians in the audience who have seen lots of case studies where you see this geographic atrophy lesion get bigger and bigger and bigger, and their Snellen acuity is exactly the same. So obviously using a 150-year-old test is probably not the way to go for a 21st century medicine. I'm watching lots of new ways where they do virtual reality microperimetry and lots of other heads-up displays, which may give us a better understanding of what the patient actually sees that we can't seem to measure. Low luminance acuity, low luminance visual deficit, all these other, what they call the soft studies that are usually secondary endpoints, as they start getting more and more validity, I think it'll move it higher up. But I think we have to look at geographic atrophy differently than the way we look at wet AMD.
So thanks, Michael. So what about, Caroline, from a drug delivery standpoint, what can you do to help us so we don't have to inject as often? What's on the horizon for this approach with the existing agents and how we can extend delivery?
Well, what we have learned, looking back the last 20 years, is when you do get an approval, you can't stop and smell the flowers for even a week. You have to start working on the next thing. And I think that, even, for example, Syfovre is approved every 8 weeks. Even that for patients in their 90s, it's a burden. I couldn't imagine even ourselves going to the doctors every 8 weeks. It would be very difficult to find the time. So I think that the next thing will be to involve other mechanisms, or to involve a systemic treatment to supplement the ocular effect. And also, as Michael was saying, to really have something for physicians and patients that ties function to anatomy. And, BCVA, vision is more than BCVA, right? And not just GA, IRDs. So we need to really tackle visual function and get something that's more durable for our patients for these diseases.
I appreciate it, and I agree, but hopefully we can package it into something that we've heard about today from the other presenters. Last comment, PK, before we wrap up?
Well, I mean, I think that GA is a very interesting case example, right? We have 2 drugs, 4 to 8 week intervals. This is a sustained release panel, so hopefully we'll have a sustained release or maybe even gene therapy for dry AMD. Yep. But the next question is, GA is too late. There's no question that the complement inhibitors should be delivered earlier. Yep. Yet getting that approved is going to be very difficult with the current rules. We have to come up with better, validated outcome measures that we can go to the FDA and say, look, we want to do this in intermediate AMD and prevent GA from forming in the first place.
Agree, and thank you all for the panel. Thank you for your comments. The presenters earlier was fantastic. We learned a lot. Thank you all.